Chapter 6 Anatomy of Flowering Plants. You can very easily see the structural similarities and variations in the external morphology of the larger living organism, both plants and animals. Similarly, if we were to study the internal structure, one also finds the several similarities as well as differences. This chapter introduces you to the internal structure and functional organization of higher plants. Study of internal structure of plants is called anatomy. Plants have cells as their basic unit. Cells are organized into tissues, and in turn the tissues are organized into organs. Different organs in a plant show differences in their internal structure. Within angiosperms, the monocots and dicots are also seen to be anatomically different. Internal structures also show adaptations to diverse environments. 6.1 The tissue system. We were discussing types of tissues based on the types of cells present. Let us now consider how tissues vary depending on their location in the plant body. Their structure and function would also be different on the location. On the basis of their structure and location, there are three types of tissue system. These are the epidermal tissue system, the ground or a fundamental tissue system, and the vascular or a conducting tissue system. 6.1.1 Epidermal tissue system. The epidermal tissue system forms the outermost covering of the whole plant body and comprises epidermal cell, stomata, and the epidermal appendages, the trichomes, and the hairs. The epidermis is the outermost layer of the primary plant body. It is made up of elongated, compactly arranged cells which form a continuous layer. Epidermis is usually single layered. Epidermal cells are parenchymatous with a small amount of cytoplasm lining the cell wall and the large vacuole. The outside of the epidermis is often covered with a waxy thick layer called the cuticle, which prevents the loss of water. Cuticle is absent in roots. Stomata are the structures present in the epidermis of the leaves. Stomata regulate the process of transpiration and gaseous exchange. Each stoma is composed of two bean-shaped cells called guard cells which enclose stomatal pore. In grasses, the guard cells are dumbbell-shaped. The outer wall of the guard cells means away from the stomatal pore are thin, and the inner walls means toward the stomatal pore are highly thickened. The guard cell possesses chloroplast and regulate the opening and the closing of stomata. Sometimes a few epidermal cells in a vicinity of a guard cells become specialized in their shape and their size are known as subsidiary cells. The stomatal aperture guard cells and the surrounding subsidiary cells are together called stomatal apparatus. Figure 6.1. The cells of the epidermis bear a number of hairs. The root hairs are unicellular elongations of the epidermal cells and help absorb water and minerals from the soil. On the stem of the epidermal hairs are called trichomes. The trichomes in the shoot system are usually multicellular. They may be branched or unbranched and soft or stiff. They may even be secretory. The trichomes help in preventing water loss due to transpiration. 6.1.2 The ground tissue system. All tissues except epidermis and vascular bundles constitute the ground tissue. It consists of a simple tissue such as parenchyma, collenchyma, and sclerenchyma. Parenchymatous cells are usually present in a cortex, pericycle, pith, and medullary rays. In the primary stems and roots, in the leaves and the ground tissue consists of a thin-walled chloroplast containing cells and is called mesophyll. 6.1.3 The vascular tissue system. The vascular system consists of a complex tissues, the phloem and the xylem. The xylem and the phloem together constitute the vascular bundles. Figure 6.2. In dicotyledonous stems, cambium is present between phloem and xylem. Such vascular bundles, because of the presence of cambium, possess the ability to form secondary xylem and phloem tissues and hence are called open vascular bundles. In the monocotyledons, the vascular bundles have no cambium present in them. Hence, since they do not form the secondary tissues, they are referred to as closed. When xylem and phloem within a vascular bundle are arranged in an alternate manner along the different radii, the arrangement is called radial, such as in roots. In conjoint type of vascular bundles, the xylem and phloem are jointly situated along the same radius of the vascular bundles. Such vascular bundles are common in stem and leaves. The conjoint vascular bundles usually have the phloem located only on the outer side of the xylem. 6.2 Anatomy of dicotyledonous and monocotyledonous plants. For a better understanding of a tissue organization in roots, stem and leaves, it is convenient to study the transverse sections of the mature zones of these organs. 6.2.1 Dicotyledonous root. Look at the figure 6.3a. It shows the transverse section of the sunflower root. The internal tissue organization is as follows. The outermost layer is the epiblema. Many of the cells of epiblema protrude in the form of unicellular root hairs. The cortex consists of several layers of thin-walled parenchyma cells within intercellular spaces. The innermost layer of the cortex is called endodermis. It comprises a single layer of barrel-shaped cells without any intercellular spaces. The tangential as well as radial walls of the endodermal cells have a deposition of a water impermeable waxy material suberin in the form of Casparian strips. Next to the endodermis lies a few layers of thick-walled parenchymatous cells referred to as a pericycle. Initiation of the lateral roots and vascular cambium during the secondary growth takes place in these cells. The pith is small and inconspicuous. The parenchymatous cells which lie between the xylem and phloem are called conjunctive tissue. There are usually 2 to 4 xylem and phloem patches. Later, a cambium ring develops between the xylem and phloem. All tissues on the inner side of the endodermis, such as pericycle, vascular bundles and pith, constitute the stele. 6.2.2 Monocotyledonous root. The anatomy of the monocot root is similar to the dicot root in many respects. Figure 6.3b. It has epidermis, cortex, endodermis, pericycle, vascular bundles and pith. As compared to the dicot root, which have fewer xylem bundles, there are usually more than 6 means polyarch xylem bundles in the monocot root. Pith is large and well developed. Monocotyledonous root do not undergo any secondary growth. 6.2.3 Dicotyledonous stem. The transverse section of the typical young dicotyledonous stem shows that the epidermis is the outermost protective layer of the stem. Figure 6.4a covered with the thin layer of cuticle. It may bear trichomes and few stomata. The cells arranged in multiple layers between epidermis and pericycle constitute the cortex. It consists of 3 subzones. The outer hypodermis consists of a few layers of collenchymatous cells just below the epidermis, which provide the mechanical strength to the young stem. Cortical layers below hypodermis consist of rounded, thin-walled parenchymatous cells with the conspicuous intercellular spaces. The innermost of the cortex is called the endodermis. The cells of the endodermis are rich in starch grains, and a layer is also referred to as a starch sheath. Pericycle is present on the inner side of the endodermis and above the phloem in the form of semilunar patches of sclerenchyma. In between the vascular bundles, there are few layers of radially placed parenchymatous cells which constitute medullary rays. A large number of vascular bundles are arranged in a ring. The ring arrangement of vascular bundles is a characteristic of a dicot stem. Each vascular bundle is conjoint, open and with the end arch protoxylem. A large number of rounded parenchymatous cells with a large intercellular spaces which occupy the central portion of the stem constitute the pith. 6.2.4 Monocotyledonous stem. The monocot stem has the parenchymatous hypodermis. A large number of scattered vascular bundles is surrounded by a sclerenchymatous bundle sheath and a large conspicuous parenchymatous ground tissue. Figure 6.4b. Vascular bundles are conjoint and closed. Peripheral vascular bundles are generally smaller than the centrally located ones. The phloem parenchyma is absent and the water-containing cavities are present within the vascular bundles. 6.2.5 Dorsiventral means dicotyledonous leaf. The vertical section of the dorsiventral leaf through the lamina shows 3 main parts, namely epidermis, mesophyll and vascular system. The epidermis, which covers both the upper means adaxial epidermis and the lower surface means abaxial epidermis of the leaf, has a conspicuous cuticle. The abaxial epidermis generally bears more stomata than the adaxial epidermis. The latter may even lack stomata. The tissue between the upper and the lower epidermis is called the Mesophyll. Mesophyll, which possesses chloroplast and carry out the photosynthesis, is made up of parenchyma. It has two types of cells, the palisade parenchyma and the spongy parenchyma. The adaxially placed palisade parenchyma is made up of elongated cells which are arranged vertically and parallel to each other. The oval or a round and loosely arranged spongy parenchyma is situated below the palisade cells and extend to the lower epidermis. There are numerous large spaces and air cavities between these cells. Vascular system includes vascular bundles which can be seen in veins and the midrib. The size of vascular bundles are dependent on the size of the veins. The veins vary in a thickness in the radiculate venation of the dicot leaves. The vascular bundles are surrounded by a layer of thick-walled bundle sheath cells. Look at the figure 6.5a and find the position of the xylem in the vascular bundle. 6.2.6 Isobilateral or monocotyledonous leaf. The anatomy of the isobilateral leaf is similar to that of the dorsiventral leaf in the many ways. It shows the following characteristic differences. In an isobilateral leaf, the stomata are present on both surfaces of the epidermis and the mesophyll is not differentiated into palisade and spongy parenchyma, figure 6.5b. In grasses, certain adaxial epidermal cells along the veins modify themselves into large empty colorless cells. These are called bulliform cells, which when the bulliform cells in a leaves have absorbed water and are turgid, the leaf surface is exposed. When they are flaccid due to water stress, they make a leaf's curl inwards to minimize the water loss. The parallel venation in monocot leaves is reflected in the near similar sizes of vascular bundles except the main veins, as seen in the vertical sections of the leaves. Summary. Anatomically, a plant is a plant made of different kinds of tissues. The plant tissues are broadly classified into meristematic, means apical, lateral and intercalary, and permanent, means simple and complex. Assimilation of food, its storage, transportation of water, minerals and photosynthesis, and mechanical support are the main functions of the tissues. There are three types of tissue system: epidermal, ground and vascular. The epidermal tissue system are made up of epidermal cells, stomata and epidermal appendages. The ground tissue system forms the main bulk of the plant. It is divided into three zones: cortex, pericycle and pith. The vascular tissue system is formed by the xylem and phloem. On the basis of presence of cambium, location of xylem and phloem, the vascular bundles are of different types. The vascular bundles form the conducting tissue and translocate water, minerals and food material. Monocotyledonous and dicotyledonous plants show marked variations in their internal structures. They differ in type, number and location of the vascular bundles. The secondary growth occurs in most of the dicotyledonous roots and stems. Thank you.